Bismillahirrahmanirrahim. Welcome to our channel. The topic which we are discussing today is Mendel's laws of inheritance. The main points that we will discuss in this topic are genetic terminologies, introduction, pattern of inheritance, laws of inheritance, law of dominance with example, law of segregation with example, and law of independent assortment with example. In order to learn about Mendel's laws of inheritance, you people have to know about some of the terminologies that are used further. First one is gene. Gene is also called as the factor. It is the unit of hereditary that contains specific characters. All the characters that are present in our body, they are represented by specific genes. For example, eye color, hair color, or any disorder like diabetes, hereditary diseases, all these are represented by specific genes. For example, here I represented capital T and small t. This is actually capital T is the gene for tall height and small t is the gene for short height. Allele is the alternate form of genes. When we represent genes in the form of pair, then it is called as allele. For example, capital T, small t, and capital T with small t. Next one is genetics. Genetics is the branch of biology that deals with study of genetic characters from parents to their offsprings. Means in this branch we study about genes, genetic makeup of an organism, and the pattern through which it is transmitted from one generation to another generation. F1 stands for first filial generation. The first generation that comes to us after first cross, it is called as the first filial generation. F2 stands for second filial generation that comes after the F1 generation. Then P1 generation. P1 is the first parental generation. The first cross that we made between two organisms is called as the P1 generation. P2 generation is the second parental generation. Usually, F1 generation is used as the P2 generation. Means F1 offsprings are used as a second parental generation. Homozygous, homo means same. So when we are using the term homozygous, it means that we are using the same alleles for cross. For example, capital T and small t. Both of them are same. So these are homozygous. Next is heterozygous. Hetero means different. So heterozygous term is used for different alleles like capital T and small t. Here capital T and small t, these are different alleles, so it is called as heterozygous. Next one is phenotype. The P of phenotype is for physical appearance. For example, when we see a flower, a red colored flower, then this red color is representing the physical appearance of that flower. At that time, we don't know about its genetic makeup. We only know about its red color, so it is the phenotype. Genotype, the G of genotype is for genetic makeup. On seeing the genotype of that red colored flower, we can know about the genetic makeup of that flower, that either it is containing homozygous or heterozygous characters or containing the dominant or recessive characteristics. So it can only be understood by genotype, studying its genotype. Next one is the Punnett square. Punnett square is also called as the checkerboard, which we use for representing the crosses. Introduction. Gregor Mendel did great work in the field of genetics, I must say fruitful work. Therefore he is called as the father of genetics. He chose pea plant for his study. The botanical name of pea plant is Pisum sativum. He chose pea plant because of its many good characteristics. He studied about the inheritance pattern of various traits and characteristics of these pea plants and its transmission from one generation to next generation. Inheritance. Inheritance is simply the process of transfer of genetic information or characters from one generation to the next generation. The characters may be good or bad. Good characteristics means like blue eye color, brown hair color, these are counted as good characters. And bad characters means the disorder or disease, like the hereditary diseases, diabetes or color blindness. These are counted as bad characteristics. So either it is a good character or bad character, they must be transmitted from their parents to their offspring, and it is called as inheritance. It is just like inheriting property to children. Just as children get part in property of their parents, in the same way they also get part in characteristics of their parents. Either these characteristics are good or bad. Inheritance pattern. Inheritance pattern describes the term how, means how the genetic information is transmitted from one generation to the next generation. There are two types of inheritance pattern. One is direct transmission and other is the indirect transmission. Direct transmission is the transmission from one generation to the other generation, means from first generation to the second generation. For example, if grandfather has eye color of blue, so it is directly transmitted to its next generation, to its son or daughter, and they also have the same blue eye color. This is the direct transmission. But in case of indirect transmission, if grandfather has blue eye color, it is not transmitted to the second generation. It is not expressed in the second generation. Rather, it is expressed in the fourth generation. Means second and third generation do not express that blue eye color. It means they are carrying, the second and third generation, they are carrying that blue eye color in the form of carrier. Therefore, the eye color of the forefather or the grandfather, it is expressed in the fourth generation. This is the carrier transmission or indirect inheritance pattern. Let's discuss example of inheritance. Here there is mother on one side and father on the other side. Mother has blue eye color with brown hair color and father has brown eye color with black hair color. Also there is represented the chromosomes. XX chromosome is for female and XY chromosome is for male. If we take the chromosomes of mother or the genes of mother as dominant character and the characters of father as recessive, then in this case during meiosis the chromosomes get reduced, means only the haploid number of chromosome, only single chromosome, they get participated in cross or mating. So in this case, if there is one X chromosome from mother and one X chromosome from father, then due to this XX chromosome, there will be birth of baby girl. And she has the same characters just like her mother, means same eye color and same hair color. Same is the case with baby boy. If this X chromosome of female, it get crossed with Y chromosome of father, then due to this cross XY chromosome, it will give rise to a baby boy, and it has the same eye color and same hair color just like his mother. Similarly, if we take father chromosome as dominant over mother chromosomes, so in this case X chromosome from father, it get crossed with X chromosome of the mother, so XX chromosome give rise to a baby girl, but she has the characteristics which are similar to that of her father, means brown eye color and black hair color. In the same way, one Y chromosome from father and one X chromosome from mother, means X and Y chromosome, it give rise to baby boy with same characteristics just like his father. This is the pattern of inheritance in which father and mother chromosomes, first they are separated, and after getting separated, these chromosomes then participated in mating, and as a result offsprings are produced which contain the same characteristics just like their parents. Gregor Mendel presented three important laws of inheritance. First one is law of dominance, second is law of segregation, and third is law of independent assortment of chromosomes. First one is the law of dominance. As the name shows, dominance means here we will talk about the dominant character. For example, in a heterozygote condition, two characters, two alleles are present together. One is dominant and the other one is recessive. The dominant is represented with a capital letter and the recessive allele is represented with a small letter. In the presence of a dominant character or the dominant allele, the recessive one will not be expressed. For example, if you take a box and there are 50 balls in that box, 40 balls are of black color and 10 balls are of white color, then in this case, 40 black balls, they are dominant, they are prominent in that box. The 10 white balls, they are not expressed. They are suppressed by the 40 black balls. Same is the case with characteristics or genetic makeup. In the presence of dominant character or dominant trait, the recessive traits are not expressed in the next generation. Let's discuss height of a person as an example of law of dominance. Here you can see in this diagram variety in height of people. Height is a polygenic character, so it is controlled by many genes. Height of humans is controlled by many genes, therefore there is variety in height of every person. For understanding the law of dominance, we will not discuss the variety of heights. We will take only two heights. One is short height and other one is tall height. Here capital T is representing the dominant character which is for tall height, and small t is representing the recessive character which is for short height. In homozygous condition, the first one is capital T, which is in this case both of these alleles are dominant, so they will express the tall height. In second case, there is small t, both of them are recessive, so it will express the short height. We can say that first one is homozygous dominant and second one is homozygous recessive character. The law of dominance is applicable in heterozygous condition when we have different alleles. One is dominant and the other one is recessive. In this case, the capital T that is dominant, it will suppress the small t that is recessive. As a result, the height of the person will be tall and we call it as heterozygous dominant character. Another example is the color of apple. As shown in this diagram, there are 3 apples. 2 are of red color and 1 is of yellow color. The red color is representing the dominant character and yellow one is the recessive character. These colors are represented by alphabet A. Capital A is for dominant and small a is for the recessive one. Through studying its genotype, you can get knowledge about the genetic makeup of the apple. First one is capital A. Both of them are dominant. So in this case, the color of apple will be red. It is called as the homozygous dominant character. In second case, both of them are small a. Therefore, due to the presence of these recessive characters, the color of apple will be yellow and is called as the homozygous recessive character. The law of dominance is applicable in the third case, where both the dominant and recessive characters are present. Dominant capital A and small a, both the heterozygous gametes are present. In this case, the dominant A will suppress the recessive A. Therefore, the color of the apple will be red. This is called as the heterozygous dominant character because this dominant A, it suppresses the small a. This is all about law of dominance. Second law is the law of segregation. Segregation means to separate. Law of segregation is also called as the law of purity of genes because the genes get separated and they independently and randomly participate in mating. Therefore, it is called as the law of purity of genes. Law of segregation states that every organism has 2 alleles per trait and these alleles they separate during meiosis. So each gamete gets only 1 allele, either it will be dominant or recessive. It means that every organism has specific traits and these traits are represented by specific alleles. So during the process of gamete formation or gametogenesis, these alleles get separated, and from diploid state they get converted into haploid condition. These alleles then individually get participated in the process of mating. The law of segregation involves only monohybrid crosses, which means cross of only a single trait or single character. We will discuss height of a person as an example of law of segregation. You can see parental generation, capital T small t in this diagram. This starting one is for father and the second one is for mother. During the process of gamete formation or gametogenesis, the genes of the parent, they get separated. Capital T, it gets separated from small t, and this capital T is separated from small t. And the gametes are formed, which is called as the gametogenesis. So this separation of the alleles or genes, it is actually called as the law of segregation. Then these gametes, they then get crossed and take part in the formation of F2 generation. If you have any ambiguity related to this law, then you have to understand this video clearly. Here there is homozygous dominant parent and the homozygous recessive parent. This first one is the parental generation, P1 generation. Then the process of gametogenesis occurs, in which gametes are formed, capital T from this one and small t from this one. These are the gametes. Then cross is made between these two gametes, and the offsprings produced will have the genotype of capital T small t, which is heterozygous. So these are heterozygous dominant offsprings. This is the F1 generation. Then again cross is made between the F1 generation. The offsprings of F1 generation, they get mated, which is called as the self-mating. These are the two offsprings from this F1 generation. The F1 generation acts as P2 generation. Again, gametogenesis takes place. The alleles of these parents, they get separated. Capital T is separated from small t. This capital T is separated from small t. Again, these are the gametes of P2 generation. Cross is made between these gametes of P2 generation, which is shown in the form of checkerboard. On one side, we will take the paternal gametes, and on other side I will take the maternal gametes. Then cross is made, capital T in one block, capital T small t in another block, small t capital T in the third block, and small t in the fourth block. These are the offsprings of F2 generation. So this is all about the crosses. Now we will show the genotype and phenotype of these generations. Genotype means the genetic makeup of the offsprings. As we can see in this F2 generation, genotype is capital T, 2 small t capital T, and 1 small t. So the genotypic ratio of law of segregation will be 1:2:1. Among these four members, one is capital T, 2 are capital T small t, and 1 is small t. This is the genotypic ratio. In the same way, we will also express the phenotypic ratio. Phenotypic ratio means the physical appearance. This capital T is for dominant, homozygous dominant. So one is homozygous dominant. So it means that this offspring will be tall. It will be tall in height. Second one is capital T small t. This will be heterozygous dominant. Heterozygous dominant, which means that this offspring will again be tall in height. Third one is small t small t. This small t is homozygous recessive. Small is for recessive, so this offspring will be short in height. So through this we can make a guess that one is tall in height with homozygous, two are tall in height with heterozygous, and one is short in height. So among the four, three are phenotypically tall in height and one is short in height. So the phenotypic ratio of the law of segregation will be 3:1. Third law is the law of independent assortment, which states that when two or more characteristics are inherited, the genes assort independently during gamete production. Which means that during the process of gametogenesis, when gametes are formed from the parents, these genes the genes of the gametes, they get separated according to the law of segregation. Now these genes and these gametes, they have to be fertilized with one another. So according to the law of independent assortment, it's not necessary that the genes of the first parent, they fertilized with the genes of the second parent in a respective way. For example, it's not necessary that the first gene of the first parent or father, it must be fertilized with the first gene of the second parent or mother. They assort randomly with each other. So this is the law of independent assortment. And second thing is that it involves dihybrid process. Means that in this law we will study about the process of two traits simultaneously, not a single trait like segregation. In order to understand the law of independent assortment of chromosome, let's discuss a general example. As we know that human beings have 46 number of chromosomes, so here there's father with 46 chromosomes and mother with 46 chromosomes, which are diploid in number. Gametogenesis occurs and there is gamete formation. According to meiosis, the number of chromosomes, they get reduced. 46 number of chromosomes get reduced into 23 chromosomes. Means diploid number of chromosomes, they are converted into haploid number of chromosomes. You can differentiate them on the basis of their colors. Same is the case with mother. Now according to the law of segregation, both the genes of a parent, they get separated from one another. Father and mother, they get separated from one another, so this is the law of segregation. Now applying the law of independent assortment, we have to make cross between the genes or the gametes of both the parents. So as a result, we would have four cases. In first case, we have to make cross between the first gene of father and the first gene or first gamete of mother. In second case, the cross is made between first gamete of father and the second gamete of mother. In third case, cross is made between second gamete or second gene of father and the first gamete of mother. In fourth case, second gene of father is crossed with the second gamete of mother. So these are the four cases according to the law of independent assortment. This is the reason that gender of the babies cannot be decided by their parents. It is a natural process and occurs randomly. As dihybrid crosses are involved in independent assortment of chromosomes, so here we will take seed shape and color together and make crosses between them. Here capital R is dominant and is for round shape, while small r is recessive and is for wrinkled shape, while capital Y is dominant and is for yellow color, whereas small y is recessive and is for green color. Just like law of segregation, We will study law of independent and assortment of chromosomes through this video for better understanding. The only difference is that we have taken only one trait of height of a person in case of segregation, but here we will take two traits together, that is seed color and shape. This is homozygous dominant parent and this is homozygous recessive parent, and they will make P1 generation. As a result of gametogenesis, the gametes are formed, capital R, capital Y on one side and small r, small y on the other side. Then a cross is made between these two gametes and F1 generation takes place. This F1 generation will be heterozygous dominant because it is containing both the dominant and recessive characters. These are the four offsprings of P1 generation. We will meet a cross between these offsprings, which is called as the self-cross or self-breed. Two of the gametes act as the P2 generation. They made P2 generation. Then the same way gametes are formed from these two parents. These gametes will be different from the P1 gametes because P1 gametes are containing only the homozygous character, while P2 gametes contain both dominant and recessive characters. As there are two characters in case of independent assortment, so it is difficult to make the gametes. So here I will tell you how to make gametes, how to make cross between them. Take one character, capital R on one side and small r on other side, and cross it individually with the other character. Capital R multiplied by capital Y, capital R multiplied by small y on the other side. Similarly, small r multiplied by capital Y and small r multiplied by small y. These are the gametes. Now we will take these gametes in the Punnett. On one side there will be gametes of one parent and on the other side there will be gametes of other parent. And then we will make cross between them in one order. Capital R capital Y multiplied by capital R capital Y, capital R small y multiplied by capital R capital Y, capital R small r capital Y capital Y, capital R small r capital Y small y, capital R capital Y small y, capital R small y. I hope it will give you a better understanding of making crosses. In the same way, capital R small r capital Y, small r capital Y and small r capital Y, small r capital Y small y, capital R small r capital Y small y, capital R small y small y, small r capital Y small y, and in the end small r small y small y. All these 16 offsprings, these are the F2 generation. They all make up the F2 generation. Now, in order to know about the phenotypic ratio, we will mark each of the offspring according to their genotype. Here I am marking capital R and capital Y offsprings with 1 to 3 numbers. The offsprings that are containing capital R with small y, just like this, I'm marking them with Roman letters. These are the Roman numbers. Then small r and capital Y with ABC. And last one, small r, small y, this one. This is the only one. Now, the phenotypic ratio of independent assortment of chromosomes will be 9 ratio, which is containing round and yellow, capital R, capital Y. 3 offsprings are round and green, capital R, small y. 3 offsprings are wrinkle and yellow, small r, capital Y. And 1 offspring is wrinkle and green, small r, small y. So among these 16 offsprings, we get the ratio of 9 ratio, 3 ratio, 3 ratio, 1. This is the phenotypic ratio of law of independent assortment of chromosomes. Here we will discuss some MCQs related to genetics, which are often asked in tests and interviews. First one is, what is true about blood type? The options are type A is dominant or type B is dominant or type AB is dominant over both type A and B, or type A and B are dominant over type O. The correct answer is option D, type A and B are dominant over type O. The reason behind this is that blood group O is a recessive blood group, whereas blood group A, blood group B and blood group AB, these are dominant over blood group O. The blood group O is represented with allele small I, while blood group A, B and blood group A and B, these are represented with alleles IAB, IA and B. A man with hemophilia marries a woman who has normal hemoglobin and is not a carrier. Which of the following is true? The options are, none of their children will have the disease, nor they will be the carriers. All boys will have the disease. All girls will have the disease. Or all girls will be the carriers. The correct answer is option D, all girls will be the carriers. The reason is that hemophilia is an X-linked disease. That is why we represent the hemophilic father with XX and Y, while the normal mother is represented with XX alleles. Then gametogenesis takes place and the gametes are formed. XX and Y are the gametes of father, whereas XX are the gametes of mother. Then a cross is made between mother and father, which is represented in the form of Punnett square. The mother gametes are represented on one side and father gametes on the other side. Then a cross is made between them and F1 generation is formed, or simply F generation is formed, which are the offsprings of these parents. The girls or daughters have the genotype of XXH, whereas the boys or sons have the genotype of XY, which shows that girls are the carriers because they have normal or the dominant X in their allelic form, XXH, which means they are not affected, they are just the carriers, whereas boys are normal. That is why the correct option is girls, all girls are the carriers. If a man is colorblind and his wife is a carrier, which of the following is true? The options are, each girl has 50% chance of being colorblind, no girl will be colorblind because girls cannot be colorblind, all boys will be the carriers and all girls will be the carriers. The correct answer is option A. Each girl has 50% chance of being colorblind. Colorblindness, or the red-green colorblind disorder, it is a hereditary disease and is X-linked. That is why we represent the colorblind father with XY chromosomes and the carrier mother with XX chromosomes. After gametogenesis, here are the gametes which are formed. These gametes are then represented in the form of Punnett square. On one side there will be the gametes of carrier mother, and here the gametes are of colorblind father. A cross is made between them, and the 4 types of offspring will be produced. In one case, there will be girl which is affected with XY chromosomes. In second case, there will be girl which is carrier with XXY chromosomes. In third case, boy is affected with XY chromosomes. And in the last case, there will be boy which is normal with XY genes. Taking into consideration the above options, girls have 2 chances. One is being affected and the other is being carrier. So the girls have 50% chance of being colorblind. Next one is a test cross is done to. Options are determine if cross has occurred, prevents sex-linked traits from being passed on, determine if an organism exhibiting the dominant phenotype is carrying a recessive allele for that trait, or prevents crossover from occurring. The correct answer is option C. Determine if an organism exhibiting the dominant phenotype is carrying a recessive allele for that trait. It means that test cross is used to determine the phenotype and genotype of the offsprings, and also to know whether the offspring is phenotypically and genotypically dominant or recessive. A farmer planted 1000 seeds of corn. The offsprings were 544 tall with yellow seeds, 188 tall with green seeds, 183 short with yellow seeds, and 64 short with green seeds. What were the genotype of parents? Options are capital T, capital T, capital Y, crossed with small t, small y, small y. Second option, capital T, capital Y, crossed with capital T, capital Y, small y. Option C is capital T, capital Y, crossed with small t, small y, small y. And option D is capital T, small y, crossed with small t, small y, small y. The correct answer is capital T, capital Y, crossed with capital T, capital Y, small y. Farmer planted 1000 seeds of corn, among which 544 have genotype of capital T, capital Y, 188 have capital T, small y, 183 have small t, capital Y, and 64 have small t, small y. So in order to find the phenotypic ratio, we will divide all these numbers with the smallest number, that is 64. Here we have divided all these numbers with 64. Number that comes are 8.5, 2.9, 2.8 and 1. Rounding of the figure, 9 ratio, 3 ratio, 3 ratio 1, which is the phenotypic ratio of Mendel's second law of inheritance, Mendel's law of independent assortment. So according to which the phenotype of the parents is dominant, that is capital T, capital Y, small y, capital T, capital Y, small y. These are heterozygous dominant. Blood group B phenotype contains anti-A antibodies in the serum and agglutinates any red blood cell with antigen. Options are A, B, O, A or B. The correct answer is option C, which is blood group A. Reason is because every person has opposite antibodies in his body in relation to its antigens. For example, if a person has blood group A, then he must have opposite antibodies, which is antibody B. Or if a person has blood group B, then he must have antibodies A. So in this case, a person has blood group B and he contains the anti-A antibodies in the serum. But if we provide this person with blood group A during blood transfusion, then opposite antibody B is produced. Both the antigen B and antibody B, they react with one another and agglutinates the blood, due to which clotting occurs. Chances for son or daughter in human birth are dash. The options are 3:1 between son and daughter, 1:3 between son and daughter, 1:1 between son and daughter, or none of these? The correct answer is option C, 1:1 between son and daughter, because both the son and daughter, they have equal chances of birth. Next one is the number of linkage groups in man are dash. The options are 2, 24, 46 or 92. The correct answer is option B, which is 24. Linkage group is actually the haploid number of chromosomes in an organism, or we can say that the total number of genes that are located on a single chromosome. It is the linkage group. In case of humans, there are 46 number of chromosomes, the haploid of which is 23. So there are 23 linkage groups in case of humans. But here they have asked about the number of linkage groups in man. So in case of man or males, there are 24 linkage groups because of the presence of extra Y chromosome. A man of blood group A marries a woman of blood group B and they have one child. Which one of the following statements about child's blood is correct? The options are: it could be blood group A only, it could be blood group AB only, it could be blood group A and blood group B only, or it could be any of the blood groups A, B, AB or O. The correct answer is option D. It could be any of the blood groups A, B, AB and O. There are two cases of this question. We will discuss each case individually. First is the case of complete dominance. Therefore, we represent the parents in the form of XAYA, XB, XB. After gametogenesis, gametes are formed, which are represented in this checkerboard. On one side, there are gametes of mother, and on the other side, there are gametes of father. A cross is made between them, as a result of which all the offsprings, they are carrying the blood group AB. Second is the case of incomplete dominance. So here the alleles are represented in the form of XAYO and XBXO, where O is representing the recessive character. Gametogenesis takes place, gametes are formed, which are represented in this Punnett square. Here there are gametes of the mother and here are the gametes of father. Whenever you have to find out the genotype of either parents or offsprings, you must have to represent them in the form of checkerboard. Otherwise, you will not get their genotypic ratio. Then here cross is made between them and 4 types of offsprings are produced. In case of incomplete dominance, the offsprings will have the chance to carry either blood group A or blood group B or blood group O or blood group AB. What are the phenotypes of a parent of a colorblind son and non-carrier daughter with normal color vision? The options are carrier and normal, colorblind and carrier, normal and carrier, and last one is normal and colorblind. The correct answer is option C, normal and carrier. In this case, among the parental generation, father will be normal and mother will be the carrier. Because according to the given conditions, son is affected and daughter is normal, so this is only possible when the normal X of father is transmitted to the daughter and normal Y is transmitted to the son. On the other hand, the affected X chromosome is transmitted to son and the normal X is transmitted to daughter, as a result of which the son will be affected and daughter will be normal. When expression of a biological character is observed in variable intensity, it is due to the effect of dash. The options are multiple alleles, codominance, epistasis, or polygenic inheritance. The correct answer is polygenic inheritance, because poly means many. Here many genes control the expression of a single character. Baldness is a dash: sex-related trait, sex-limited trait, sex-influenced trait, or none of these. Correct answer is option C, sex-influenced trait. A gamete without any sex chromosome is called dash. The options are nucleogamete, autosome, recombinant gamete, or all of these. Answer is option A, nulogamete. SRY gene on Y chromosome of a man determines dash: height, skin color, hemophilia, or maleness. The correct answer is option D, maleness, because SRY gene is the gene that is only present on Y chromosome, and Y chromosome of a person represents maleness. All of the following are examples of X-linked recessive disorders except dash: colorblindness, diabetes insipidus, hemophilia, or Alport syndrome. The correct answer is option D, Alport syndrome, because Alport syndrome is an autosomal disorder which is related to somatic cells, while the other three, that is colorblindness, diabetes, and hemophilia, these are X-linked diseases. How many different kinds of gametes will be formed by an individual who is heterozygous for 4 gene pairs? Options are 8, 16, 20, or 30. The correct answer is option B, 16. The reason is that when we talk about 2 gene pairs, as in the case of law of segregation, here we took square of that 2. 2 to the 4, so in that case 4 types of gametes are formed. But in that case, we are talking about 4 gene pairs, so here we take square of 4. 4 4 is 16, so the answer is 16. 16 different types of gametes are formed in this case. Next one is, a woman with normal color vision whose father was red-green blind married a red-green colorblind man. What is the probability of her firstborn child being red-green colorblind? Options are 1.0, 0.75, 0.50, or 0.25. Correct answer is option C, 0.5. In this case, father is affected with color blindness, whereas mother is carrier. Mother is normal but carrier, because father of this mother is affected with color blindness, so the affected X chromosome of that father is transmitted to his daughter. Therefore, this mother is a carrier. Now, the normal process of gametogenesis takes place. Gametes are represented in this Punnett square. A cross is made between them, as a result of which 4 types of offsprings are produced, 2 of which are colorblind, 1 is a carrier, and 1 is normal. As here we are checking the probability of colorblind child, so the colorblind childs are 2. We will divide this 2 with the total number of offsprings, that is 4, and the number that comes is 0.5, which shows that the probability of colorblind child among these 4 offsprings is 0.5. Last one is, 2 parents each of blood group A have a daughter of blood group O. What is the probability that their next child has blood group O? The options are 0.125, 0.25, 0.50, or 0.75. The correct answer is option B, 0.25. Here both the parents have blood group A. Father has allelic form of XAYO and mother has the allelic form of XAXO. Both of them are representing the blood group A. Gametogenesis takes place. Gametes are formed which are represented in this Punnett square, and then a cross is made between them. 4 offsprings are produced, 3 of which have blood group A and 1 has blood group O. The offsprings containing blood group A have the genotype of XAXA, XAYO, and XAXO, whereas the blood group O is represented with genotype of XOYO. As here we are discussing the probability of blood group O, so we will divide this 1 offspring with the total number of offspring 4. The number that comes is 0.25, which shows that the probability of blood group O child is 0.25. This is all about Mendel's laws of inheritance. If you still have some problem related to this topic, then you can ask in comments, and please don't forget to subscribe and like our channel. Thank you for your time.